All right, let's talk about a mystery that's been puzzling scientists for decades: Parkinson's disease. You might know it as a condition that affects movement and coordination, but beneath the surface, something much stranger is happening inside the brain. And recently, scientists have uncovered a brand new clue that could change the way we think about this disease. So, at the center of this story is a protein called alpha-synuclein. Normally, this protein helps brain cells communicate, but in Parkinson's, it gets a pretty bad reputation. For years, researchers thought the biggest problem was that alpha-synuclein clumps together, forming toxic blobs that disrupt everything around them. But it turns out there's a sneakier villain at work: alpha-synuclein oligomers. Now, what are these oligomers? Imagine them as smaller, rogue gangs of alpha-synuclein that don't just sit around clumping up. Instead, they go on the attack. Researchers from Aarhus University in Denmark discovered that these oligomers can poke tiny pores, yes, actual holes, in the membranes of brain cells. It's like your cells are being sabotaged from the inside. Here's how it works. The oligomers start by attaching to the cell membrane. Then, they begin to push themselves partly inside, and finally, they create a pore, a little tunnel, right through the membrane. Through these pores, molecules that should stay inside the cell start leaking out, and things that don't belong can slip in. This chemical chaos could be a major reason why brain cells start to malfunction in Parkinson's. But here's the twist. These pores aren't just passive holes. They open and close again and again. So, the cell isn't doomed right away. Its own internal pumps might keep things stable for a while, but it's fighting a losing battle. And it gets even more interesting. The oligomers seem to prefer attacking curved membranes, the kind you find on mitochondria, the tiny power plants inside our cells. This could explain why energy production in brain cells breaks down in Parkinson's patients. Now, you might be wondering, can we stop these rogue proteins? The researchers have already tested special nanobodies that can spot these oligomers, but for now, they don't stop the pores from forming. Still, it's progress. Of course, there are still big questions. We don't know exactly why alpha-synuclein turns from helpful to harmful in the first place, and it's not clear if this pore poking behavior happens the same way in living brain cells. But every new clue brings us closer to understanding and maybe even stopping Parkinson's disease. So next time you hear about Parkinson's, remember, the real battle might be happening at the tiniest level, with proteins playing both hero and villain. Stay curious.